Alright, continuing on with 15.3 and linkage. I'm going to pick up where we left off with our two parents who were a wild type heterozygous female with a doubly mutant homozygous male. Keep in mind that we expected most of the offspring to be heterozygous or homozygous, and we also saw that this was because these two alleles were linked on the same chromosome from mom, and these two alleles were linked on the same chromosome from dad. So we couldn't have this particular parent passing on that little b with that VG plus too often, and we couldn't have this parent passing on this BP plus along with this VG too often because they're on different chromosomes. So moving along with that idea, how exactly did we get those 206 and 185 or so flies that did not have this genotype or this genotype? We weren't expecting that. We were expecting a 1-to-1 to 0-to-0 ratio because of the linkage. Hopefully you're starting to put the idea of crossing over and meiosis in your heads. So looking at Morgan's experiment again, we can see that the little b and VG genes are located on the same chromosome. Looking at the F1 fly that was crossed with the double mutant shows us the F1 was heterozygous, and the B plus and the VG plus alleles were on the same chromosome. And conversely, the B and VG were on the opposite homologous chromosome. So here are those two parents, and we can see that once these two chromosomes made a duplicate of one another, we have a duplicated chromosome consisting of two sister chromatids, and a duplicated chromosome consisting of two sister chromatids. Occasionally, these regions of the chromosomes would cross over, and as a result, this VG plus on the red chromosome would end up bound to the white chromosome or the pink chromosome, and then the VG allele would be bound to the red chromosome. So we have a little bit of crossing over, which resulted in a recombinant chromosome, which we see right here. It's recombined. Now we have B plus VG and B VG plus on the chromosomes. Notice on this parent, it didn't matter if crossing over would occur or not. A VG is going to be swapped out to a VG, and it wouldn't change the chromosomes at all. So keeping in mind that there weren't a whole lot of these recombinants, most of our flies were of the parental nature, either this parent or this parent. The genotypes were the same. How close are these two genes actually, in terms of how close are they located to one another on the chromosomes? Well, we can figure that out by looking at the recombination frequency, or how often these two alleles are recombined during gamete formation. To do that, we need to look at the total number of flies produced, and we need to look at how many of those total flies produced showed the recombinant genotype. So if we add all those numbers up, we get 2,300, and if we add up the number of flies that showed the recombinant genotype, we would get 391. So 391 divided by 2,300, and then multiplying that by 100 to put that decimal into percent form, we get 17%. This number 17%, keep in mind 1% equals 1 map unit. We now have identified the gene right here and the gene right here to be 17 map units apart, meaning that they show recombination 17% of the time. So the lower the number, the closer the two alleles are together, the two genes are together, and the less likely you are to get recombination. the further apart they are from one another, the higher percent of recombination, the less linkage they show. So once this idea was starting to be understood with linkage of genes on the same chromosomes, geneticists started to construct genetic maps, which more or less just shows of the genes on the same chromosome how close their loci are. So the locus here and the locus here are 48.5 map units away from one another, or they show recombination 48.5% of the time. So this happens to be looking at those flies. You can have the antennae essentially, which is the iridae, black body, the eyes, vestigial wings, normal wings, so on and so forth. It was Alfred Sturtevant that constructed the genetic map. He happened to be one of Morgan's students, and his hypothesis was that the further two genes are away from one another, the higher the probability that crossing over will occur, hence, or resulting in a higher number of recombinant alleles that we would see in the phenotypes. This shows us three particular genes, 1, 2, 3. The distance between these two is 17%, and the distance between these two is 9, and the distance between those two is 9.5%. Why 9 and 9.5 doesn't add up to 17, I don't quite get it. But then I guess it could just be due to chance of these alleles having a higher recombination for various reasons. Maybe just there's a certain spot that crossing over tends to happen more for some unknown reason. So a few slides ago I had mentioned that 1% recombinant frequency equals 1 map unit. Here you actually have it in a little bit of writing now. So when we saw that recombination showing 17%, that was equaling 17 map units according to Sturtevant's mapping system. Genes that have a higher frequency of recombination than 50%, so they're more than 50 map units apart, are considered to be genetically unlinked, even if they are physically on the same chromosome. And the reason for that is that crossing over will occur between chromosomes, and those alleles will be separated at about the same frequency as if they were on two totally different chromosomes. In other words, they're so far apart that they're not even really considered to be linked. So if it's over 50%, they just exchange quite frequently, so they're not linked, is what that's saying. So at this point, we're done with 15.3. We'll have a few more videos moving through chapter 15. Looks like there's about 9 slides left, so we're almost done with this chapter.